Hello everyone. Thank you for joining us in celebrating APrON's 10 years. Congratulations APrON study, and may you continue for another 10 years and more. I am Brenda Leung. I am an associate professor in the Faculty of Health Sciences at the University of Lethbridge. I am a co-investigator with the APrON study and have been so since its inception. I remember quite well working on the original proposal for APrON, and like a parent, had hopes for its potential, but not really knowing how far or long it would go. So I applaud Dr. Maternal and the team for leading APrON to where we are today. I'm so pleased to be here and to share some of APrON's research on maternal mental health. The idea for APrON started with two main health concerns: mood disorders in the perinatal period and developmental disorders in the offspring. While maternal mood was a primary outcome of interest from the onset of APrON, maternal mental health, broadly speaking, was also of concern as a potential predictor or exposure associated with pregnancy complications, that is, poor outcomes, as well as infant and child development. We recognize the importance of maternal mental health when we first did a review on the role of nutrition and perinatal depression. This article was published in 2009, and it has been one of the most highly cited articles in the journal formerly known as the Journal of the American Dietetics Association. In the review, we reported that inadequate intakes of certain nutrients, such as the omega-3 fatty acids, folate, B vitamins, iron, and calcium, which we would typically see in a standard Western diet, puts women at increased risk for depression, especially in the perinatal period. And we know that poor maternal mental health leads to poor physical and emotional behavioral development in the offspring. So poor maternal mental health affects not just the individual, but also their families and friends, as well as long-lasting personal, social, and economic impact. And you can see from this figure here some of the outcomes as a consequence of maternal depression for the women during pregnancy in terms of birth complications, as well as for the infant. Maternal mental health was assessed by several standardized psychometric instruments at multiple time points. For symptoms of current depression, we use the Edinburgh Postnatal Depression Scale. And for anxiety, we use the Anxiety Subscale of the Symptoms Checklist 90. Stressful life events was measured using the Modified Stressful Life Events Questionnaire. And for the most common DSM-4 Axis 1 disorders, we used the Psychiatric Diagnostic Screening Questionnaire. We also asked moms about their level of social support using questions from the StatsCan National Population Health Survey, as well as their perception of their general health using the short form 8. Now, I will present the research or the studies we did on maternal mental health. I have grouped them into 4 areas. First, the studies done on the impact of maternal mental health on children's development. 2, lifestyle and socio-demographic factors on mental health. 3rd, maternal mental health and physiological response. And then the last group is the reliability of psychometric measures used in a pregnant population. We have 4 studies that investigated maternal mental health on child development and health. The first study of adverse childhood experiences seen in parents was found to be transmitted to the offspring by way of mother's depression and anxiety. It is known that moms who report having ACEs are more likely to have symptoms of anxiety and depression during the perinatal period. This is found to be associated with children with increased externalizing problems such as aggression, noncompliance, and oppositional behavior. Furthermore, boys seem to be at increased risk to the indirect effects of maternal ACEs by both anxiety and depression. So this may explain the mechanism by which adverse childhood experiences are transmitted intergenerationally. The second study looked at both maternal and paternal perinatal depressed symptoms and whether that has an impact on child's behavior at age 2 or 3. And the results show that moms with depression, and when both parents have depression during the perinatal period, that is associated with internalizing behavior in the children. When only moms have depression, then we see increased likelihood of externalizing behavior, that is the aggression, attention problems in their children at age 2 to 3. So for the findings of this study suggest that healthcare providers should consider the whole family when treating perinatal depression. The third study looked at maternal prenatal anxiety and how that impacts children's externalizing and internalizing behavioral problems. Prenatal anxiety is predictive of postnatal anxiety, and that can interfere with the security of the maternal-child attachment and may further increase the risk of child behavioral problems. This study found that prenatal anxiety was associated with both externalizing and internalizing behavior, but only in children with an insecure style of attachment. Secure attachment means that the bond between mother, parent, and child meets the child's need for security, calm, and understanding. So attachment security can reduce the likelihood of children's externalizing and internalizing behavior as a consequence of prenatal anxiety. The fourth study here looked at maternal depressed symptoms both prenatally and postpartum and how that may affect brain structure development in the children. And what was found is that there is reduced thickness in children's premature developed brain when it is exposed to higher maternal perinatal depressed symptoms. Therefore, ensuring optimal women's mental health throughout the perinatal period is important to ensure optimal brain development in the child, because non-optimal brain development can affect tasks involving inhibition, attention control, and executive function, and in the long run can affect cognitive and behavioral outcomes throughout the child's life. So these findings suggest a possible mechanism by way of maternal perinatal depression that increase the vulnerability of the children's susceptibility to depression later on in life. This next group of studies looked at lifestyle factors on maternal mental health. One of our earlier publications investigated micronutrient supplementation and postpartum depression. Women who reported higher scores on the APrON postnatal depression scale reported having lower intakes of supplements. This is especially in the case of selenium and the omega-3 fatty acids. They also reported that those with higher EPDS scores were more likely to be born outside of Canada, had more chronic conditions, had more stressful life events during pregnancy, lower social support, and higher prenatal depressive symptoms. The second study examined sleep quality and mood during the perinatal period. Most women, not surprisingly, reported having sleep disturbances during the perinatal period. However, women who had the worst sleep quality and those who had more increase in sleep problems throughout their pregnancy reported having the highest levels of anxiety and depression. And they also had the lowest social support. And in addition, those who had poor sleep during pregnancy were also more likely to report having depression in the postpartum period as well. The third study looked at socio-demographic background of both parents. And the purpose of the study was to look at factors associated with postpartum depression in partnered parents. The results show that postpartum depression in both mothers and fathers were predicted by low household income, high prenatal depressive scores, low social support, and higher numbers of stressful life events. And for fathers in particular, smoking was also predictive of postpartum depression. So when we compare couples who were not depressed, those that reported postpartum depression in both the mothers and the fathers, they were more likely to have low income, higher prenatal depressive symptoms, and low social support. Next, we have a couple studies that looked at physiological factors in maternal mental health. This first study is interesting because it looked at psychological distress and cortisol levels within the same individual during pregnancy. So within individuals, it was found that the greater the negative mood, the higher cortisol levels were within the same individual. So it looks like the levels of psychological distress co-vary with cortisol levels over time. And these findings may support the proposal that there is a plausible biological mechanism between distress and cortisol levels, and explains the relationship between maternal experience and fetal development. The second study examined maternal cardiac vagal control, or CVC, and how that is associated with maternal depression and gestational hypertension. So CVC reflects the input of the parasympathetic branch of the autonomic nervous system, which is involved in cardiac regulation. And the study found that the greater the depressive symptoms were, the lower the CVC was in pregnancy. And this was proposed to be indirectly associated with gestational hypertension. And this could further lead to pregnancy and birth complications. So these are some interesting proposed mechanisms behind maternal mood. And lastly, we have a couple studies that examine the use of psychometric tools in evaluating maternal mood and anxiety. The first one looked at the Psychiatric Diagnostic Screening Questionnaire, which has been used in the general population, but this was one of the first times that the PDSQ was administered to a pregnant population. And so interestingly, the PDSQ found that social phobia, obsessive compulsive disorder, somatic disorder seemed to be higher in the pregnant cohort when compared to the literature. And this may reflect more of the changing physical and mental status of our pregnant cohort than an actual reflection of an increase in those disorders. Also, there seems to be poor agreement between the PDSQ and the other measures of depression using the EPDS or anxiety using the SCL-90 anxiety subscale. So what this tells us is that the PDSQ subscales may actually not be appropriate for the pregnant population, and further research is needed into more appropriate instruments to determine the psychiatric disorders that may be in the pregnant population. The second study examined the anxiety scales that were used in the APrON cohort and compared it to another cohort called All Our Babies. When the women from both cohorts were compared, they were similar in a number of sociodemographic characteristics. The women tend to be under age 35, more likely to be married or in common law, largely Caucasian, and more than half were having their first child. However, there were low correlation amongst the 3 anxiety subscales that were examined. It seems that the scales did not measure anxiety as a single dimension, and therefore they were not comparable in how they may have conceptualized anxiety. So it seems that these scales, again, are not suitable for measuring anxiety in the pregnant population. And that concludes the work so far on maternal mental health from the APrON study. But many more questions remain, and there is more work to be done. We are currently analyzing blood nutrient levels and its association with maternal mood, and we hope to have a paper completed soon. So any interest in the APrON data by other investigators is always welcome. Thank you very much for your attention and your interest in the APrON study. And so now, if there are any questions, I'm happy to answer them. Thank you.